almost over, okay? And you started watching it at the beginning, and so you're just going to stick with it until the end, right? You're not going to, I mean, you always hate to like, oh, I only watched half of that series, you know, I binge watched half. So, okay. Mechanisms of hormone action. There has to be a receptor present in the target cell. The question is, where is the receptor going to be? So far in this class, the place where we see receptors the most is at the membrane of the cell. And the receptor location is external. So that the intracellular fluid, the intercellular fluids, the fluids that pass between cells, okay, they can encounter the membrane of the cell and the receptors present. Hormone is going to be delivered to the environment around the cell, and then the hormone can encounter receptors at the membrane of the cell. That's one way. Plasma membrane receptors are the type that we see with the water-soluble type of hormone, which is amino acid derivatives, polypeptides, proteins. If it helps, once we get into the polypeptide size and the protein size, cells can't make holes big enough to allow those to pass through. Okay? And oftentimes our amino acids that get built into these proteins are sort of hydrophilic. So they're more water soluble than they are fat soluble. So the plasma membrane's phospholipid core is a problem. It rejects them. So they don't have any open holes to go through, and they can't go through the membrane's lipid layer. So they interact with receptors on the surface. On the other hand, the lipid-soluble types, the steroids and some amino acid-based hormones that are lipid-soluble, those things pass through, even if they're big, they pass through the lipid membrane no problem. And so they don't interact necessarily with receptors on the surface because they can get into the cell so readily. So the receptors for these kinds of hormones, like steroids, is inside the cell itself. Cytoplasm or nucleus. And if the receptor is in the cytoplasm, then the hormone interacts with the receptor in the cytoplasm and then it moves to the nucleus. Or the hormone goes direct to the nucleus where its receptor's there. Now what's going to happen next then is that this receptor and a hormone complex is going to lead to the copying of genes in the nucleus and the making of proteins. Okay? This is the method or the action of water-soluble hormones that have their receptors at the membrane of the cell. It's a long list. It uses a series of internal messengers. It is very much like the G protein mechanism. It's just there are going to be some differences from other G protein mechanisms that you've seen before. Okay? We have a new player. This is a common player when it comes to hormone receptors. Now, its explanation is very similar to the GMP explanation that I gave you earlier. The P is for phosphate, the M is for mono, the A in this case is for adenosine, which is similar to adenine, which is built into DNA. This one is cyclic AMP. The molecule, I couldn't even tell you what it looks like, but obviously the C in front of us tells us that it has some kind of a cyclic structure. Okay? Hormone interacts with the receptor. Okay? The receptor activates a G protein. The G protein its shape changes, and then it fits or it's able to bind with an inactive enzyme. Okay? In this case, we have the enzyme called adenylate cyclase. This is similar to what we saw before with the eye and the photoreceptor. So we have an inactive enzyme ready to rock as long as we take this G protein and stick it into a binding site on that adenylate cyclase. Adenylate cyclase then converts ATP, which we know is around in the cell, to cyclic AMP. Cyclic AMP. This is an internal messenger now. The hormone's an external messenger did not come into the cell. Instead, it activated a process that makes cyclic AMP. Cyclic AMP then diffuses in the cell, where it interacts with another inactive enzyme called a kinase. A kinase is an enzyme which phosphorylates proteins. In this class, you have seen the effect of phosphorylating proteins. It causes the protein to change its shape. It can take an inactive protein and activate it, okay? And those inactivated proteins become active, and then they can participate in a chemical reaction of some sort, okay? As long as the hormone is fitting into the receptor location, this process will occur. Hormones bump into receptors and then they bump out. Just like any other molecule, it's constantly moving. If it bumps into a receptor, it turns on this process and then it diffuses out of the receptor location. If there's a lot of hormone around, then it will bump in right again and continue the process. But if the hormone release drops, then the amount of bumping into the receptor location declines and the effect of the hormone declines. So it is very much then based on chemical concentration of the hormone in the blood as to the amount of effect. Okay? It's not quite threshold and action potential kind of concept, but it is saying that you've got to get your hormone levels up to a certain level in order to have the desired effect. And as soon as those hormone levels begin to decline, then the desired effect begins to go away. This is saying here then that cyclic AMP, once it is produced, it is being destroyed. So if we don't fit the hormone in, then the cyclic AMP eventually gets destroyed in the cell, and the cell goes back to its pre-condition. Intracellular enzymatic cascades have huge amplification effect. What this means is, if you know a little bit of enzyme chemistry, when you turn on an enzyme, that thing just rocks. It works very quickly, and as long as that enzyme is active, it is catalyzing a reaction, and that reaction is going and going and going. Okay? So if you turn on 10 enzymes, which then go and lead to the turning on of another kind of enzyme, then you're going to really amplify the system. If I can get it across to you well, if I need this enzyme turned on, okay, in order to make another messenger that can turn on another enzyme, if I turn on 10 of this enzyme, then I can turn on hundreds of another. If I need him to be active in order to make another messenger that activates another enzyme, if I make 10 of him, then I'm going to be able to amplify this system. And that's what happens. To put it all together, these water-soluble hormones bring about changes very rapidly. And they can be very significant changes in a short period of time. The problem is that they can also bring these changes down fairly rapidly. Now, when I say rapid, I don't mean milliseconds now, okay? Milliseconds is action potentials. The endocrine system is working on the seconds, not milliseconds. So it can come up quickly, but not as quickly as a nervous impulse. What you just saw listed there is similar to what you see here, although now we're seeing a different player. We're seeing this player known as GTP, okay? We're adding in. Don't get too nervous about it. It's the same concept. And then I think we'll be done. There's our receptor. This is a amino acid derivative, a polypeptide, or a protein. It is in the fluid surrounding the cell, and it bumps into the receptor location. In this case, what we see is that the G protein activates, okay? We're seeing that the receptor is leading to a change in the G protein so that the G protein can then go and activate adenylate cyclase. Adenylate cyclase will take existing ATP that is present in the cell and convert it to cyclic AMP, which can now serve as a second messenger. Cyclic AMP can now activate protein kinases, which can then activate other enzymes, or they can phosphorylate a protein and turn it into an active enzyme or something along those lines. Okay? So this is the mechanism for your water-soluble hormones. In general, there are exceptions. All right, well, that should do it then for now. When we return, we will finish up the endocrine system and probably have time for review, discussion, whatever.